Hello, welcome to Biology, the scientific study of life. Hardy-Weinberg equilibrium. Suppose that the individuals in a population not only donate gametes to the next generation at random, but also mate at random. That is, all male-female matings are equally likely. Not only will this population have the same allele frequencies from one generation to the next, but its genotype frequencies can be predicted from the allele frequencies. Such populations are in a state of Hardy-Weinberg equilibrium. It turns out that our wildflower population is in Hardy-Weinberg equilibrium, as this video notes that using the rule of multiplication, we can calculate the frequencies of the three possible genotypes assuming random unions of sperm and ova. The probability that 2c to the power r, notice that, alleles will come together is 0.8 by 0.8 equal p by p equal p to the power 2 equal 0.64. Those about 64% of the plants in the next generation. We will have the genotype c to the power r, c to the power r, the frequency of c to the power w, c to the power w. Individuals will be about 0.2 by 0.2 equals q to the power 2 equal 0.04 or 4%. C to the power r and C to the power w heterozygotes can arise in two different ways. If the sperm provides the C to the power r allele and the ovum provides the C to the power w allele, the resulting heterozygotes will be 0.8 by 0.2 equal 16% of the total. If the ovum provides the C to the power r allele and the sperm the C to the power w allele, the heterozygous offspring will make up another 0.8 by 0.2 equal 16%. We can summarize this unions of gametes in an algebraic equation. Notice that allele frequencies of male gamete between two brackets p plus q between two brackets p plus q allele frequencies of female gametes equal p to the power 2 plus 2 pq plus q to the power 2 genotype frequencies in the next generation. This represents as with allele frequencies, all of these genotype frequencies sum to 1. Thus, the equation of Hardy-Weinberg equilibrium states that at locus with two alleles, the three genotypes will appear in the following proportions. Notice that notice p to the power 2 plus 2 pq plus q to the power 2 equal 1. This is the equilibrium. If a population were in Hardy-Weinberg equilibrium and its members continued to mate randomly generation after generation, allele and genotype frequencies would remain constant. The system operates somewhat like a deck of cards. No matter how many times the deck is reshuffled to deal out new hands, the deck itself remains the same. Aces do not grow more numerous than jacks, and the repeated shuffling of a population's gene pool over the generations cannot in itself change the frequency of one allele relative to another. Note that a population does not need to be a Hardy-Weinberg equilibrium for its allele frequencies to remain constant. Many species, such as the bees that Mundell used in his experiments, do not mate randomly, since bee pollen normally matures before the flower opens. It fertilizes the same flower that contains it. Mendel could cross his pea plants only by artificial fertilization. Because of self-fertilization, pea populations are far from a Hardy-Weinberg equilibrium. All the homozygous plants produce only homozygotes, while about half the offspring of the heterozygous plants and homozygous. It does not take many generations before almost all the plants in the population are homozygous, but even though such species do not mate randomly, they still produce their gametes at random from their gene pools without other influences. Their allele frequencies will not change over time, just as populations that do mate at random. Genetic variation, the raw material for evolutionary change, is presented even in the self-fertilizing peas. Conditions for Hardy-Weinberg equilibrium. The Hardy-Weinberg theorem describes a hypothetical population that is not evolving, but in real populations, allele and genotype frequencies do change over time. This is because the following 5 conditions for non-evolving populations are rarely met for long in nature. 1. Extremely large population size. The smaller the population, the greater the role played by chance fluctuations in allele frequencies from one generation to the next, known as genetic drift. 2. Gene follow. The transfer of alleles between populations can alter allele frequencies. 3. No mutations, by introducing or removing genes from chromosomes or by changing one allele into another. Mutations modified by the gene pool. 4. Random mating, if individuals preferentially choose mates with certain genotypes, including close relatives. In breeding, random mixing of gametes does not occur. 5. No natural selection. Differential survival and reproductive success of individuals carrying different genotype will alter allele frequencies. Departure from these conditions usually results in evolution, while natural populations rarely, if ever, are in a true Hardy-Weinberg equilibrium. In many populations, the rate of evolutionary change is so slow that these populations appear to be close to equilibrium. This enables us to obtain approximate estimates of allele and genotype frequencies, as the following example shows. Population genetics and human health. The Hardy-Weinberg equation can be used to estimate the percentage of the population carrying the allele for an inherited disease. For example, about 1 out of every 10,000 babies in the United States is born with phenylketonuria, PKU, in metabolic disorder that results from homozygosity for a recessive allele. Left untreated, PKU results in mental retardation and other problems. Now born are now tested for BQ, and their symptoms can be lessened with phenylalanine-free diet. To use the Hardy-Weinberg equation, we must assume that people do not choose their mates on the basis of whether or no they carry this gene, and do not generally mate with close relatives in breeding. We must also neglect any effects of gene follow from other populations into the United States. Introduction of new PQ mutations and differential survival and reproductive success among PQ genotypes. These assumptions are reasonable since inbreeding in the United States is not common. Populations outside the United States have PQ allele frequencies similar to those seen in the United States. The mutation rate for the PQ gene is low and selection occurs only against the rare homozygotes. If all these assumptions hold, then the frequency of individuals in the population born with PQ will correspond to Q to the power 2 in the Hardy-Weinberg equation, Q with to the power 2, which means a frequency of the homozygotes for this allele. Because the allele is recessive, we must estimate the number of heterozygotes rather than accounting them directly as we did with the pink flowers, since we know there is one PQ occurrence per 100 or 10,000 pairs, which means Q to the power 2, which means one part of thousands, one part of 10,000s. The frequency, notice that the frequency of recessive allele for PQ, which means equal which equal 1 to the power 100. And the frequency of the dominant allele is P equal 1 minus Q equal 1 minus 1, 1 part of 100 equal 0.99. The frequency of carriers heterozygous people who do not have BQ but many pass the BQ allele to offspring is 2BQ equal to by 0.99 by 0.01, which means 1 to the power 100 equal 0.198, approximately 2% of the US population. Remember, the assumption of Hardy-Weinberg equilibrium results in only an approximation. The real number of carriers may be somewhat different. Nevertheless, we can conclude the many harmful recessive alleles at this and over like are concealed in the human population because they are carried by healthy heterozygotes.